Okay, now the third part of the lecture 7. We jump to plant genetic transformation. This is a very important part of the plant biotechnology. We had discussed in the previous chapter about a number of techniques that are used for making recombinant proteins and using bacteria, using different types of cutting machineries. For example, we used a number of restriction and nucleases, making the staggered cuts or the blunt ends, and then we had also discussed about the adapters and the DNA linkers. All those things were so that you can have a base of genetic engineering. Now we're jumping towards a little more sophisticated because there we were talking only about prokaryotes, but now we're talking about genetic engineering in eukaryotes, higher plants. So the transformation, genetic alteration of the cell resulting in the direct uptake or incorporation of exogenous genetic material from its surrounding and taken up to the cell membrane. It could be direct method or it could be indirect method, which means we are actually introducing new genes into the plant genome. All these, there are a number of genes already present in the plant, but we are introducing some new genes into the plant, and we call them plant genetics transformation, genetic engineering plants, transgenic plants, all these methods. Now there are two methods. Number one is direct and number two is indirect. What is meant by direct methods of plant genetic transformation and what is meant by indirect method, we will discuss in the next few seconds. Now, when we say transformation methods, what do we exactly mean? For example, this is a plant gene. You see, this is a plant gene. We can transform. So transform means you can have a paired piece of DNA entered to the cell, or you could actually use tungsten particles, DNA coded. This DNA is coded on these particles, and you bombard it with a projectile gun, and these particles get inside. And when they get inside, you hope that some of these particles will make their way and integrate the DNA coded on these particles into the main genome. It says tungsten, but I can tell you in the previous times, tungsten was used. Of course, tungsten is really expensive. It's not expensive, sorry. You can use gold as well. And in the previous times, people used to have micro scale particles because it was not so easy to make nanoparticles. But these days, most of this microprojectile gun thingy for plants is through nanoparticles of gold, because gold is really, really good. Tungsten can have some toxicity while gold have no toxicity. And microinjection. Microinjection means you have a suction pump. You have what do you call it? You have pipette, micropipette. You can actually get inside a plant cell or any cell, not only plant cell. You can get in, if you want to do microinjection in a plant cell, before doing this experiment, you will have to digest the cell wall, because the cell wall is the rigid outermost boundary of a plant cell, and without digesting or getting rid of the cell wall, you can never enter. So for this method, for microinjection, you would have to use enzymes, for example, cellulase. They would actually solubilize, they would get rid of all the plant cell wall that is composed of the carbohydrate cellulose. And you can inject through a pipette, and this could be aided from live microscopy. So you are actually seeing where is your pipette going, and you could get inside a cell. There's some DNA inside your pipette, and you can slowly push it in the nucleus. Now, when you do this, you hope for two things: that the injury that you have caused this cell by injecting the micropipette is not beyond recovery. The cell can recover itself. At the same, if you want to inject it inside the nucleus, in this case, it's a prokaryotic cell. There is just a prokaryotic cell in the figure. But if you're working with a plant cell, it would not be a prokaryotic cell. There will be a double membrane bound nucleus. So you have to get inside the nucleus. And then you would hope that the nuclear membrane, as well as the plasma membrane or cell membrane of the plant cell, it can actually regenerate itself. Beyond recovery, it's not beyond recovery. And then you would hope somehow this piece of DNA has found a way and got incorporated into the genome, the chromosome of the plant, and also getting expressed so that the gene that you have inside it is also expressed, and either the product or any other goal that you have through these transgenics is fulfilled. You can regenerate the cellulose of any plant cell. I mean, you have a plant cell with the cellulose, you digest the cellulose, you do your manipulations, and then you can regenerate the cellulose cell wall again. It's not a big, it's not that difficult. So when we talk about direct biolistics, particle bombardment, as we already discussed here. So you have the DNA coated on the nanoparticles of gold or tungsten or microparticles, and you place them in front of your plant cells and you shoot them, and you hope that some of these particles would go and reach the nucleus, and the DNA on them would dislodge and it would get integrated into the genome. And it works. It works like magic. The efficiency is very low, but it does work. Electroporation is another method, the same for plant cells as we discussed in bacterial cells. You give high voltage shocks to the cells and it creates pores inside the cell membrane. The DNA pieces can enter and then it can go and incorporate into the cells. Polyethylene glycol is also used for these purposes, and microinjection. Polyethylene glycol is mostly used in protoplast culture. Protoplast culture is when you get rid of the, what is a protoplast? A protoplast is a plant cell without cell wall. So if you treat a cell wall with cellulase enzyme, it will dissolve the cell wall of the plant, and then you will have just a loose mass of the plant cell that is known as protoplast. And when you want to do protoplast culture, so you have a number of plant cells, and you dissolve their cell walls, and you want to mix them so that their nuclei mix and a hybrid kind of plant is developed. You use polyethylene glycol in that culture, and it somehow protects the cell protoplast from bursting, as well as it aids in making two cells come to each other, unite, and then in between the cell membrane is dissolved and the two nuclei mix. Microinjection we already discussed. So this is a typical process for biolistics or particle bombardment, or we can also say microprojectile or macroprojectile. And these DNA coated microcarriers, as I discussed a few minutes ago. And these days, all this work is done at the nanoscale. And these are not no more microparticles, but they are nanoparticles, mostly gold. Why? Because gold is noble, does not have any reaction, it does not have any toxicity. Electroporation, as we already discussed, there's some DNA lying around, you can see, and the cell is happy. But when you give it electric shocks, it gets disturbed. There are a hole created in the, there's a hole, you can see, cell membrane is ruptured at places, and the DNA can insert and in saying hi. And then you hope that this cell membrane somehow repairs itself, and your DNA that is inside, if it's not a prokaryotic cell, gets its way into the genome of the plant and incorporated inside the chromosome. Indirect method is agrobacterium tumefaciens. Now, agrobacterium tumefaciens is an important topic that we will discuss in great detail tomorrow, but just a small introduction about agrobacterium tumefaciens. It is actually the causative agents of crown gall in tobacco plants, also known as what do you say? Tumor. It's crown gall. It's caused a tumor. You can see it's like a tumor, a big bulb of irregularly uncontrolled cell divisions of the tissues of plants. So also known as plant tumor. I'm going to study tomorrow. And this is very important because in Agrobacterium tumefaciens, there is a plasmid which is known as TI plasmid. So TI means tumor-inducing plasmid. And inside the TI plasmid, there's a region very important which is known as tDNA. tDNA means transfer DNA. And this is the main factor how this Agrobacterium tumefaciens would cause the disease, how it would make a plant tumor inside tobacco plants. This TI plasmid and then this tDNA. And we can exploit this for plant genetic engineering in a very efficient way that we will discuss in the next lecture tomorrow. So take care of yourself, watch the video with great care, and bombard me with questions. Until then, I would say bye and happy learning. Allah Hafiz.